I would like to welcome May Griffith, professor at the University of Montreal, who just gave a very interesting lecture about biomaterials and cornea transplants. And now this is kind of the Q&A part, which again, we don't have an external audience, but I am recording because I will put it on our YouTube channel. And I was very happy to see that you have a lot of questions on Slido. So that's great. We'll get to a point where I'll leave it to the audience to ask the questions. But we thought it's a good idea to just have a short conversation about May's career path. And basically, she's been on a couple of continents. She's been a professor at 3 different universities. So maybe you can tell us a little bit, from your beginnings in Toronto to Sweden and back to Canada.
Okay. Yeah, so I'm one of these people who doesn't really like to move. I did my undergrad, I did my master's, and I did my PhD at University of Toronto. And then I couldn't stay.
What was your major, by the way? Sorry.
Oh, I did cell developmental biology. So I started off with biology. And then I actually spent about half a year at University of Alberta doing a postdoc. And that was the time that it took for me to submit my thesis and to defend my thesis. So after the first winter in Edmonton, I decided it was time to go to Boston. So I went to Harvard Medical School and did a postdoc there. And when I was at Harvard Medical School, I got recruited for junior professor position as an assistant professor at University of Ottawa.
Were you already involved in clinical trials by then? Is that something that's been from the beginning of your academic career?
No. I worked in very much a basic science lab, although at the time, Bjorn Olsen, one of my co-supervisor, postdoc co-supervisor, was involved in a new company at the time, Organogenesis, and they wanted to make corneal implants, as organ on a chip, but it was not organ on a chip. It was huge constructs. And so I thought that was kind of cool to make artificial organs in a dish for testing purposes. So that's what I did. But when I got to University of Ottawa, I was at the Eye Institute. And one of the first things I did was to try and set up my wound healing program. I called up the eye bank and I said, I want to look at wound healing. And do you have donor corneas that I could use for my research? And the lady at the eye bank basically laughed at me. She goes, "Are you crazy? Don't you know that there's a huge shortage of corneas?" So, okay, that was a no-go.
And sorry, I'll interrupt because it raises other questions. An eye bank. So there are actually eye banks.
Yes, eye banks, they collect tissues. So eye and tissue banks. Yeah.
Okay. Now, is this something that's just on paper? You know where they are, or are they actual eyes located in
There's
boxes?
an actual eye bank. The main eye bank is in Quebec.
Okay.
Yeah. But we have a small one at our hospital, Maisonneuve-Rosemont. And yes, if you look in their fridge, you will actually see their corneas or full globes, in the containers in the fridge.
Wow. So that means that even a full globe you could reuse, like you can just transplant an entire.
Well, no. There are a few people who are trying to do that. Right now, they're transplanting bits.
I see.
Mostly the corneas.
I see.
Yeah.
I see. Cool. Fascinating.
Yeah. So anyway, I couldn't get corneas for research. I had problems with using rabbits because of the animal facility rules at the time. So the technicians have to put all the drops in. And what I was trying to do was to track my cells. So I was putting a fluorescein drop that would, label my cells, and I could follow them during the wound healing. So the issue is with the eyes, technicians can do two things. I tell them each of the right eyes needs a drop. So some of them will actually give the rabbit's right eye to drop, right? They stand behind a rabbit, or they know it's that eye, but other people will stand in front of the rabbit, use the right hand, drop it. So you can see my right hand is pointing to your left eye. 50% of the time the rabbit's got the drops in the right eye that I, and then 50% of the time it was in the wrong eye. So I could see my very, very first grant go down the toilet. This is not good. So I remember what I was doing in my postdoc, for the last few months before I left, which was making artificial corneas. So I said, I'm going to make one of those. And of course, when you make something for testing, the director at the Eye Institute at the time is a cornea surgeon. And he goes, why are you wasting your time making things for testing cosmetics when there's so many people who need cornea transplants? So he decided for me at the time that I was going to make corneas for transplant. So that's how it started. And I've been doing it ever since.
Yeah, so this is University of Ottawa. How does Sweden come into the equation?
Oh, well, this is lovely Health Canada. We made our implants and we were ready to bring them into clinical trials. And so my boss, like I mentioned, who's a cornea surgeon, was ready to test this. So went off to Health Canada. And the first question I was asked was, so who's done this before? It's like, no one, I hope, because we want to be the first in the world. Well, then we don't know how to regulate this. So this went on. And at the time we were collaborating with the Swedish surgeon. He would fly every 3 months to Ottawa, to check the pigs. And he said, it looks like you're having problems. So the deal was we would always do the first trial in Canada and Sweden would be number 2. And he goes, well, if number 1 setting doesn't work, can we go ahead in Sweden? And so at the time, the chair of my department said, sure. So I took a sabbatical. Went there when he started his clinical trial. And one day the rector of the university invited me for lunch. And I thought, oh, that's really nice. So he said, how would you like to extend your stay? Remember, Ottawa is as cold as Montreal. The Linköping in Sweden, on a really, really cold day, you get -5. So anyway, I was there.
It's called damning with faint praise, like when you say -5 is warm, then your criteria of warm have been biased by Canada.
Yes. So, -40 is unheard of. -5 is a cold day. And they assured me that winters are normally around 0. So anyway, he said, how would you like to extend your stay? And I thought, I either go back to -40 or I stay and around 0. It was pretty much a no-brainer. So I stayed for almost a decade.
Wow.
Until I got invited by UDM to come back to put in the translational program here.
Wow.
Yeah, so it was, and the other thing that I came back was because my kids were becoming teenagers. You really, really don't want to have teenage kids in Sweden because they have no discipline. You cannot give a kid a timeout.
Wow, interesting.
The kids loved it. I tried to get my daughter to do homework. It's like just 30 minutes a day before TV. And she goes, no, and you can't make me. I know the laws. Yeah. They are actually taught their rights when they're about 5. So they all know their rights, but they are never taught their responsibilities.
That's amazing.
So as Canadian parents, we've been telling the girls, you know, you have rights, but you have responsibilities that go with the rights. And responsibilities seems to have just gone whoosh. So it's like, no, we're not going to do the 30 minutes. And so my husband got a little bit fed up after a few months of this. And he goes, yes, I can make you do homework. We're going home. So, yeah, so here I am.
Back to minus 20.
Yes, I guess there are some positive aspects to climate change, although I shouldn't be saying it.
No, but of course, you know, like it is getting warmer here.
It is
so-
getting warmer. Like I haven't actually seen minus 40, but like knock on wood, because now that I've said it, we probably get minus 40 now.
Imagine if somebody offered you a sabbatical in Florida. Would you go for that, or is that too warm?
Actually, I was offered a position in Florida before Sweden.
Okay.
The other thing is I also value my life. Don't like random drive-by shootings, and Miami is not the safest place.
So it's a trade-off between
it's a
climate
trade-off,
and other
yeah.
things. So now that you're here in Montreal, maybe you can tell us a little bit about this translational medicine program that has gotten off the ground.
Well, I was recruited to ophthalmology, so obviously they wanted the cornea program to continue. Now, we had done solid implants before when I was in Ottawa in Sweden, so they wanted something new. They wanted something big, obviously, you know, to put UDM on the map. And what could be bigger than to try and solve the problem with 13 million people needing transplants? So we definitely needed something new.
Mhm.
And so we came up with the liquid cornea program. When I first came, we did some of the work back in Sweden because it took a couple of years before my lab was ready. So we still did the animal work back in Sweden. And my first Canadian postdoc was a very, very happy person. He got to travel to Sweden to do his biomaterials, put his biomaterials in pigs, because back then we had the 2-syringe mixing system. And so you actually needed a trainee to do the mixing for the surgeon. So he happily got on a plane, went to Sweden, and he was even more happy to get on the plane to go to Australia,
Right.
where we did our rabbit study. So yeah, Chris was a very, very happy trainee. So now we have our labs and we can do our own animals here. We don't get the trainees flying all over the world like they used to.
Right. Now, you mentioned that, you know, cornea causes blindness. I'm curious, what percentage of blindness is caused by cornea problems?
So in terms of absolute numbers, I would say it's really hard to measure, because the way that the research has been done, they've gone from a measure of what is the most prevalent, and the most prevalent, of course, is cataracts, because it's the most common, and cornea, depending on whose ranking, is number 4 or number 5.
I see.
According to WHO. So I would say it depends on the country. So if you go to a country like India, it would be like your number 1 main cause. But in Canada, it's mostly age-related diseases.
I see.
Yeah.
And is this something to do with genetics, or just the way that the health system is set up?
I think the health system being set up the way it is, and also like in a country like Canada, there is not as much heavy industry. So you don't find, you know, a lot of damage from burns. I was invited to go to Chile just before the pandemic, but it was not the pandemic that was the issue at the time. It was a revolution. And during that revolution, I heard that the police were shooting at the people with rubber bullets. And so the front of the eye, which is the cornea, is all damaged. So right there and then, within weeks, you know, you have a few hundred to a few thousand people with damaged fronts of the eye. And of course, they did not have very efficient eye banking. And I think they still have issues with getting cornea transplants. So I'm working with a Chilean researcher who is a professor in Ottawa to try and, you know, still solve this problem. And had we had our liquid cornea in clinical use, we could probably have patched up the eyes of many of these patients.
Right. Now, it looks like there's been lots of creative solutions to this in the past. The one with the eye is just something I can't get out of my mind. And then you said smokers' teeth do not
Did not qualify.
work.
Yeah.
Why?
I think it's because of the nicotine.
Huh. Okay.
So nicotine has an effect on, I guess, systemic issues as well, like, you know, blood pressures and all that. And I guess the nicotine is so ingrained or embedded in the teeth because of the pores, it does something. I'm not sure what, but I guess.
Yeah, just of all the categories, you know, like
Yeah, I
street
know.
smokers from donating teeth to fix a cornea.
Right, and they use their own teeth or their own bone, so you're not using an allograft.
Right.
People are now trying to make artificial teeth to hold that PMMA, but in my mind, it's like, why go through all that problem when you can just make a, you know, a much friendlier implant,
Right.
which
Right.
you don't have these hard bits and you don't have a multiple surgery. Like the implants that we made in Sweden, it's a single surgery.
I see.
So I'm, you know, wondering about the people who are making the false teeth so that you could put the false PMMA in and do this procedure. So I think that's the other thing, is that sometimes the researchers have tunnel vision. They see the area, but they don't really see outside.
Remind me, correct me if I got this wrong, but the cornea already has stem cells. Like they are there. We all have them.
Yeah.
So this is really the reason why you can implant something, because the stem cells will eventually create
they will
the
go in, yeah.
right cells. Can those stem cells be used for other organs? Are they really just as generic as that, or are they still kind of
They are,
cornea?
most of the stem cells would be cornea based. And if you want to take them out, I'm sure there are some primitive cells, but you have to biopsy them. And then, you know, to expand them. So instead of using the cells from the cornea, people found it more effective to use the same stem cells, but found in the skin. And that's where the IPS comes from, right? The induced pluripotent cells. So you can harvest a lot more from skin than you can from tiny cornea biopsies.
All right. So these are the questions that came up during your lecture for me. Like that's what I was curious about. Students have been asking more insightful questions. So maybe we'll just go down the list of the questions that we have on Slido, and we'll start with how they're upvoted. Okay. Looks like Erwan's question was the most upvoted, and maybe Erwan, would you like to ask it, or should I read it?
Yeah, sure. I can ask it. I'm just wondering, why is it better to get farming? You mentioned it's better to get from animal origin from cornea than from cadavers.
Okay, so if you get everything from cadavers, there's a lot of regulations, right? And not everybody is going to donate their body to science. And especially that was an issue with trying to harvest cadaver collagen. Donating your body to science doesn't mean that a company can come in and get harvest the collagen. That was the main issue. Because you donate your body, you didn't sell your body. And then, you know, a company coming in using a donated cadaver to try and make a product there are a lot of ethical problems with that.
Can you tell us a bit about Quebec? I was also fascinated that Quebec does things differently with cornea donations. How is this regulated in Quebec?
I am not sure, so don't quote me on that. But I believe that they are able to harvest corneas from patients who pass on more effectively. Not sure about the laws, but even if you've signed your driver's license in most other places, they will ask your family. Yeah, so Quebec is currently self-sufficient in donations.
So it's not just cornea, we're talking just in general organ donations.
Well, I can't speak for the other organs. I'm
Right.
just aware of the corneas.
Right.
Yeah.
Right.
The other provinces will harvest, but they also purchase from some of the eye banks.
I see. I see. Thank you. There was a question from Charlotte. Would you like to ask it?
Yes.
Yeah.
So what do you think are the next steps to bring the liquid cornea to clinical use?
Okay, we have to finish our large animal study. We have to prepare a submission to Health Canada. And the next step that we're doing is actually a meeting with Health Canada. So you meet with the officials and you bring them their preliminary data. So this is called a pre-CTA meeting. So clinical testing approval meeting. So we're not actually asking for the approval. We're not even asking for a pre. We're asking for a pre-pre. So you can imagine the steps. So you do a pre-pre, then you do a pre, and then you work on all the submissions, find out what documents are missing. Mostly you would work with the regulatory consultant, and then you do your actual meeting with Health Canada to get the clinical trial authorization, or the CTA. For medical devices, it's investigational testing authorization, so an ITA. So we would do that. We would probably also do ISO 10993. We have to complete that for the portfolio. We have to do stability testing. So quite a few other tests to go before handing in the portfolio, and then they take their time to examine, and then they'll get back to us.
So this is related to Alyssa's question. Maybe you can ask it now.
Just quickly, I wanted to ask you, like, how long do you think this would take, like, time frame wise?
I'm thinking 2 to 3 years if we're really lucky.
Oh, okay.
Yeah, because you don't know when you submit something to Health Canada, what they want or what they don't want, because they don't give you much guidance. You send it to them and then they will comment. They don't tell you you need to do this, you need to do that, which makes it more difficult. So I'm more used to like the Swedish FDA or the European Medicines Agency, where you actually go there and they actually tell you what you need to do.
You had a follow-up question on Slido, so maybe now is a good time.
Basically, I mean, your liquid cornea sounds like very innovative, very creative and everything. I don't know if there were any like limits that it has right now, since we haven't talked about this.
We'll find out the limits when we finish our large animal study.
Okay.
What we think is the main limitation right now is using it on patients with very severe conditions. So, so far we've done rabbits and we've done pigs, and we've got 100% sealing, which is nice. So all the wounds will seal, and we're very happy for that. But we found out that when we did regrafts on a couple of the animals, so with the pigs, they try to scratch their eyes. When you're healing, wounds tend to be itchy. And with human patients, you can tell them don't scratch. But with the pigs, we found that at Charles River, they were trying to scratch their eyes on their metal food bowls. Why on earth would they give pigs food in metal food bowls? I don't know. And we paid them to look after our animals. We didn't expect them to let our animals scratch their eyes. Anyway, they did. So we ended up regrafting, but when we found the regrafts, the animals were neovascularized because of this injury that they had with their food bowls. And so when their eyes are neovascularized, they sealed, which was nice, and we get reepithelialization. But the implants were not as clear as the ones I showed you. They were more translucent to opaque. Whether that's going to clear in about 9 months, I don't know. We've done other animal studies before that were hazy, but they started clearing. But so the limitation could be that in patients that had vascularized eyes, we may need a second transplant. So you might just seal whatever damage there is, and then wait for the patients to clear, or if it doesn't clear, then we might need a second graft. So, you know, after it's healed, we might need to remove the opaque tissue and either retreat with the liquid cornea again, or maybe retreat with the solid implant that we make, which basically is the liquid cornea, just solidify it in a mold. Yeah, so a lot of questions we don't know yet until we finish our study.
So this goes well with Sami's question. Sami, would you like to ask it?
So, are there any technical considerations that you
We will of course have to go through ethical approval, and for clinical trial it'll be different. There are a couple of things to consider, and one of it, you know, would be I think the ethics is not so complicated right now because everything is synthetic. It's fully synthetic. We're not using stem cells. We're not using anything that comes from somebody else. So while we do have the ethics approval, all the stem cells are going to come from your own body. We don't take them out. They're going to grow in. So what we've done is we've tried to minimize the level of ethical issues. There are still going to be issues to consider, which is what does the technology do? Like in a country where you don't find, for example, transplants, what is this technology going to do to, you know, the what is going to be the social impact? What is going to be the economic impact? So we're going to have to look in terms of the ethics of doing this. So, like from my point of view, I don't see a huge issue because we're not using embryonic stem cells. We're not even using donated cells. So we're not looking at sale of you know, organ parts or something like that, because everything is artificial. But we do have to consider what is this going to do to the society, where people have been dealing with blindness in a certain way, and then now you're going to make this change, right? Whether this change is for the good or not. It's hard to imagine that it might be a bad thing, but I've spoken to researchers in London, actually at the London School of Hygiene and Tropical Medicine, where this group was just introducing glasses, just spectacles, to school children in a couple of villages. And they did not expect to see a backlash, and there was a backlash. The people who were wearing glasses were now considered different, so they, you know, they could see that they were somehow disabled. so there was bullying and all kinds of things that was happening with these people. So they would rather not wear the glasses and not even see than to use a new technology. So we have to go in with a little more sensitivity, and that's where I think the ethical considerations come into effect, because we were told that we're coming in with the Western technologies and changing their lives, not necessarily always for what they would consider better. We would consider it better, but they might not.
Right. So I think this is a good point to step a little bit, to zoom out. And there's been a couple of questions about, in general, organs on a chip.
Mm-hmm.
And there's a question from Jane and a question from Mona. I guess they're both related. Oh, sorry. It's always the same problem. We have Muna and Mona, and Mona and Muna. Okay. But to make things more complicated, Muna writes the name with M-O-U and A. And that's why I keep making that mistake. So I apologize to both of you. It's a rare occurrence, but happens in a class. So please, maybe you can ask your question.
Yes. When you said that we can stop using animal models as bloods, what if we use body on chip where we have all the organs connected and we can actually have the same reaction of the human body to that drug, especially when we use the stem cells or the patient himself?
Well, I think that's what the organ on a chip people are headed towards. So, the first organs I showed you that we're just in a dish, and now we're trying to add circulation to it. So maybe some tears, some people are trying to make blinking. So that's all good, but then there's also the systemic system. So like you said, you would pretty much have to hook up all the organs of the body, because you might not expect things to happen. But, when we did our herpes study in the rabbits, you find that the biomaterials actually have an effect on the draining lymph nodes, right? And then you would have systemic effects. So 10993, you look at systemic effects of very, very local implants. So hopefully someday the organ on a chips will get to that stage where it will mimic everything systemic. But for now, we still need systemic considerations.
Now researchers are working on making body on a chip, like the whole body on a chip.
Mm-hmm.
And I think it will be more accurate and more related to our physiology, more than the animal models, especially if it will be personalized. Like we will use the cells of the patient. So, for example, if he has a disease and we want to test a drug with him to cure this disease, we can test the drug using his cells. And that's what I find so interesting.
Right. It sounds really good. There are practical issues. Like if we did that for every patient, the costs would be astronomical. It's not viable right now. Hopefully they can shrink it to a different size, that maybe it would be body on a chip, but it would be groups of cells representing each organ. But right now it's going to be astronomical. So even with clinical trials, so our hospital runs clinical trials with stem cells. So you isolate stem cells from a patient, and you might do your gene therapy, you do the editing on the cells, put it back into a patient. It's extremely expensive. So the cost of doing something so personalized right now, it's just not going to be feasible, right? You're basically spending $15 on a mouse versus hundreds, maybe thousands on trying to isolate cells from a person. And even if you're using IPS that comes from the patient's skin, you are going to spend a lot of money with different growth factors, differentiating it into brain and eye and heart and whatever. So maybe in the future, it might be feasible, and it might look very nice for publication in Nature or Science. But then, you know, when it comes to actual clinical application, you have the real world coming in.
And I think this is related to Jane's question. So you touched on a bit, but maybe Jane can follow up.
So do you think that's the only reason why organs on chip are not gonna replace animal organs, like the price or something?
Right now it's the economics. The other thing is it's not sophisticated enough. So you can get organ on chip, and some of the organs are more advanced than others. There is no homogeneity. So I think the regulatory agencies are going to need to harmonize. So the model that's developed, for example, at University of Toronto for the heart is way more advanced than most of the other models developed in other countries. Which do you do? So if you're going to harmonize this, you know, is Health Canada going to adopt the Canadian model, or are they going to adopt, you know, the Japanese model? Which model? And then each of the regulatory agencies right now have to harmonize all the testing and the ISO standards. So it's going to be a long way before you standardize the different tests. It may happen in the future. It's going to need a lot of drive from people in the different regulatory agencies to harmonize. So Health Canada has to harmonize with EMA, and they have to harmonize with Japanese, and then the FDA always does its own thing. Yeah, which organ on a chip? For every organ in the body, it's going to be a different one. There is an organ on a chip from Spain that's also with the heart. It's maybe not advanced in certain ways, but they've actually had a whole circulatory system going through. So, you know, are these two groups going to combine? It would be great if they did, but right now, not all researchers work with each other. So I think it's going to take a much longer time than we would like to see organ on a chip being the go-to model. So ISO right now still uses animals. We hope in the future it would use organ on a chip, but the standard will need to be standardized.
But do you think it's only like either a price issue or a work issue, or is it something concerning directly the organ on a chip like that?
I think it's going to be harmonization. You're going to have to make it equal. So all the drugs would have to be treated, tested on, you know, one way. Like, for example, now you have drugs being tested on rodent models, or, you know, as I showed you, the ISO for allergies, it's always subcutaneous or intramuscular. So it's pretty much harmonized with ISO and ASTM. So you're using the same model. They tell you it's 1 centimeter away from the vertebral column. So to do an organ on a chip, which model, whose model? You're going to need to determine that before, you know, Health Canada and the EMA, they have to agree. Because the implants that are in the body, they are subjected to drugs as well. So for example, if we have a cornea transplant or implant, and actually one of our patients in Sweden had to go for a cataract surgery. So you can imagine how much panic that caused the surgeon and myself, because, you know, this patient was the first patient to have an artificial cornea, and now this woman wanted a cataract surgery. It's like, oh my God, how are the drugs going to affect her? How is the cataract surgery going to affect her? Because we now have to open another and make another opening in the eye, and then do phacoemulsification, remove her lens and put in a new lens, and then she's got eye drops. So that was huge. So thankfully, cataract surgery went well, eye drops are not a problem. But yeah, you know, it's just something that we had to remember, that she was tested on collagen-based implants. And what if the implants were not collagen-based? How would those eye drops have reacted? We don't know. Thankfully, we never had to find out.
Yeah. So you showed us a very manual operation on the cornea. Then you said Indian doctors are using lasers.
Not all Indian doctors.
Well,
at
this
the- at the
particular, this
WHO
particular
hospital.
And then Nika's question is even more general than that. So Nika, would you like to ask that?
Yeah, I have a question about if you think robot-assisted surgery can really change the errors that the doctor can make, or make cornea implant more successful or not? Or is it a concern, the way that they are doing the surgery, or it's not a concern
For the cornea, that's not a concern because it's a fairly simple surgery. And what we're doing with the liquid cornea is actually to try to make it even simpler, so that you don't have to be a cornea surgeon to do it, but to allow a medic who is working, for example, in a refugee camp, to be able to just administer and to be able to seal a wound. So that's one thing. The robot-assisted surgeries, I think those will be way more useful when you have complex surgeries like brain surgery, cardiac surgery. And I know even when I was in Sweden, they were already, doing some of that. So we tend to do more drug treatments for Parkinson's. The Swedes were using electrostimulation with electrodes. So they were implanting, and then it was sort of more AI controlled, electric stimulation. And after they stimulated, the patients were at least 3 months without Parkinsonian symptoms. Whereas here we do drugs, and we do like stem cell transplants. So yeah, it's going to be very different. And obviously the brain, you need the whole map, whereas cornea, it's not difficult to find the cornea, it's right there. Yeah.
Thank you. And then for another kind of high level question, Moritz had one.
Yeah, so this course is about emerging biomedical technologies. So I was interested in what, in your opinion, what do you think are the most promising topics in biomaterials right now, or the most emerging overhyped or not overhyped topics?
So if you go to the conferences, it's really different. Some people are promoting the use of biomaterials, and some others are promoting the use of small molecules. So it's hard to compare one to another. And what was really amazing was I was listening to a talk yesterday by a guy who's doing genome editing, doing gene transfer. So that seems to be like the big thing in biotech. And they were doing lentiviruses. But to actually hear this guy talk, and he said, oh, we're doing so much gene editing. Won't it be great to have biomaterials that will just do the work so we don't have to do gene editing? And then he said, oh, and won't it be nice to have nanoparticles instead of using lentivirus to do the gene transfers? So it seems like it's going in circles. What may be the hottest thing in biomaterials today might circle back to something else, like not using biomaterials, but using just biomaterials to make particles to do gene transfer. And then self-assembling biomaterials were really hot, like maybe a few years ago, but now it seems like everybody is trying to make particles that deliver genes or nucleic acids. And I'm sure it's going to be different again. I also spoke to the person who actually made the first 3D printed cornea implants. And I was talking to him about where he was going because everybody was so excited about 3D printing. And so he was the guy who made the first 3D printed cornea implant. And then he said, oh, it's not going to go anywhere. It's like, what? He said it was wonderful when we published this, but the reality is it's going to take an hour to print each one. And then he said, and we know that, you were taking about 5 minutes to mold one. And then we were saying, well, and I'm not even molding them anymore because now we're trying to just inject them as a liquid so that they will just solidify on their own. So really hard to predict.
But next week you will have to choose a topic. So maybe May can help us with the choice of topics. I asked her to think a little bit about how to direct you on what to cover next week. So basically, students will read a paper and they will have 3 minutes to present that paper. And they will have 3 formats to choose from, but they have to use one format. So if they use text this week, then they can't use video, then they can't use text the other week. So that makes it a bit more diverse. Okay. What should they be reading about? And maybe some keywords, interesting topics too.
Well, I think it's what they're interested in. Like because biomaterials covers a lot, from nanoparticles to deliver nucleic acids, and then what are biomaterials? Right? Go back to the definition. So DNA could be biomaterial. So if somebody wants to present on DNA origami, hey, why not?
Okay.
People are trying to build DNA-based cages to maybe deliver drugs. Other people might be more interested in titanium implants. How can you get the surface smoother? How can you make the implants less resorbable? Because we know that macrophages are actually eating away in the surface of titanium implants, and that's why some people need to change them in 5 years, some people in 15 years. In my area, trying to do injectables in corneas, yes. You can maybe do injectable in corneas, but did you know that people were doing injectables or moldables for pieces of bone in the face? There's a group in Sweden doing that. So they call it injectable. So they call it, it's not sure what the name is, but you can look it up. It starts with a cornea. It starts with a liquid. They mold pieces of bone, which is designed with CAD drawings, and then this has been used to reconstruct the skull. Yeah, it's done by a group in Uppsala University in Sweden. They have dental implants. There's also the group in Gothenburg, which they were using nanocellulose. So nanocellulose from plants, from bacteria. A group in Portugal, they're working on corneas, but they are using fish waste. So, they have a big fishing industry. And actually, I was on the PhD thesis committee of this woman who was looking at isolating collagen from fish skin for making corneal implants. And in their lab, they were also making skin implants. So you could look at that. It's very nice. How do you avoid the next environmental issue? like, you know, the Japanese releasing their radioactive waste, mercury spills, oil spills. So you could look into a very good idea, but this very good idea needs to have a few other steps. Like maybe the skins will need to be filtered to ensure that there's no contamination from heavy metals. And what do you do when you are dependent on a marine resource as a biomaterial? So marine resources, I mean, the idea that they had to use waste bone and skin and, you know, other parts, it's a wonderful idea. But, you know, there's some constraints, which is like heavy metal pollutions, oil pollution. And what can you do? I'm sure you could try and skim off all the metals, make sure that it's within the allowable range of however many parts per million. So I think the topic is broad enough that if somebody has any interest in any of these areas, it would be well worth a discussion.
Yeah. And again, that's tougher than, you know, telling you this is the topic. Really, you know, feel free to explore. Obviously, it's a very broad field. As we discussed last time, try to find papers where you can actually read something else about them. You know, just reading the raw paper itself, sometimes it's too complicated, too dense. So find a paper that has been discussed elsewhere. I think the context that we got is also important. So try to put it in context, try to find something that's exciting to you personally, and, you know, try to have that passion shine through in your presentation. Those were a lot of questions and very insightful one. Is there anybody who would like to ask something that wasn't on Slido, or maybe it was and I didn't get to see it? So Alyssa and then Ellie.
So, you had the clinical trials on artificial corneal surgeries, what do you call it, and then you moved on to liquid cornea. Are you still using like corneal implantations, artificial, like in the real life, or it's just you just completely?
Well, it's an interesting question because University of Ottawa licensed the technology to a company. So the company right now, it's US-based company. They're trying to do clinical trials, but I'm not involved in that. And so, you know, there should be these are solid implants that we'll be doing trials with. And we also are doing trials with a colleague in Spain. Hopefully that'll be soon. Like the pandemic really shut things down for a couple of years. Our clinical trials that we did were not perfect because with some patients with stem cell deficiencies, the vision was not fully recovered. So the next trial we're going to do, we're going to put, for the patients that have a stem cell deficiency as well, we're going to load the implants with stem cells and then transplant it into the patient. So this has been ongoing for a couple of years now. Right now it's with the Spanish FDA. So whenever they give the go ahead, we will be running a trial, and then the companies who've licensed the technology, of course, they will be doing I'm sure they are doing the trials right now. I just don't know at what stage it will go into clinical use.
Thank you.
Thank you. And Ellie?
I wanted to ask you because you said you use 3D printing to print molds and so on. And so did you or another thing for directly printing, like bioprinting cornea directly with 3D impression or not?
Yes, I mentioned that I was talking to the person who did the first 3D printing, and he decided that he got a nice paper, but it's not going too far. The problem is the layer by layer, right? You know how the printers work? They deposit one layer and then the other layer, and then so anything that is spaced more than 300 nanometers will scatter light. So you can see that each of the layers, they will scatter light. And it also takes about an hour to print one. Whereas if we mold it with the molds right now, so the molds are made, well, the molds we use come from a Swedish mold maker. They are not 3D printed molds. They are just injection molded molds. And it takes about 5 minutes. Yeah.
All right, so I think this is a nice place to stop. It's been almost an hour. And thank you for joining us, and thank you all for the questions. We really appreciate it.
I think it's a pleasure. It's really nice to actually have people ask lots of questions.
Yeah, thank you. So now we're gonna